For the first time, important questions on our relationship with the Neanderthal could be addressed. The time of divergence could now be determined to around 800,000 years ago. Paabo and his team also surprisingly found that gene flow had occurred from Neanderthals to Homo sapiens, demonstrating that they had children together during periods of coexistence. Importantly, comparisons could now also be made to search for differences between Neanderthals and Homo sapiens, differences that may underlie features that are unique to all humans living today. Paabo's team also retrieved DNA from a minute finger bone found in Denisova cave in the Altai Mountains in southern Siberia. Again, they started by analyzing mitochondrial DNA, and to their surprise, the sequence was different from both present-day humans and from Neanderthals. Paabo had discovered a completely new hominin entirely by analyzing and comparing genome sequences. This was a sensational discovery. The new hominin was given the name Denisova. Paabo's team next analyzed the nuclear Denisova genome sequence, enabling important investigations. Neanderthals and Denisovans were shown to be sister groups, diverging from each other around 600,000 years ago. They further found evidence of gene flow from Denisova into Melanesian Homo sapiens, indicating that mixing had occurred. Svante Paabo's groundbreaking discoveries have provided important new knowledge regarding our evolutionary history. Neanderthals and Denisovans lived in different parts of Europe and Asia for hundreds of thousands of years and adapted to these environments, while Homo sapiens evolved and lived in Africa. By mixing with them after migrating out of Africa, Homo sapiens picked up sequences that improved their chances to survive in their new environments. We now know that present-day humans in Europe and Asia can carry 1 to 2% of Neanderthal sequences, and that Asian and Southeast Asian populations carry an additional up to 6% Denisova sequences. Examples include a gene variant that improves living at high altitude in Tibetans, and sequences that affect how our immune system responds to different infections. No extinct hominin DNA has yet been sequenced from Africa, since conditions for preserving archaic DNA are poor in tropical climates. Thanks to new advanced computational methods, it has still been possible to find indications of gene flow from as yet unknown archaic hominins into Homo sapiens on the African continent.